Welcome to The Mind, the Cure Protocol for August 15, 2026. No newly verified patient reaching milestone in this pass has reached care, so we will not pretend otherwise. We are following what changed in the evidence, what new research it opens, and what still needs proof before it can help anyone. This protocol is a living loop. People bring the questions that matter. Direct evidence changes the map. The source ledger remembers what we have already seen and checks the original date before calling it new. Every signal ends with one practical next action, the experiment, validation, collaboration, or care decision that could move it closer to real benefit. Researchers reported ProteinDPO, a direct preference-optimized protein language model that aligns a structure-conditioned generative model to experimental stability data while preserving pre-trained biological knowledge. The result is a concrete step toward using foundation models not just to predict proteins but to engineer them against fitness targets, illustrating the intelligence inversion at the center of AI-driven biomedicine. Generalized models increasingly act as design engines for disease-relevant molecular functions. If confirmed, this could help drug teams test better molecular ideas sooner instead of searching blindly. The useful part is a smarter starting point, not a finished medicine. Biology still gets the final vote. Next, show with independent benchmarks and laboratory work that the targets produce better drug candidates. Researchers reported in Nature Communications a lysostaphin-based fusion protein, engineered by adding thermosensitive elastin-like polypeptides, that improves stability, lowers immunogenicity, and enables sustained release to eradicate MRSA biofilm-associated implant infections. If confirmed, this could give antibiotic teams a better starting point against resistant infections. The useful part is a more practical route to treatment. The chemistry still has to survive scale, safety, and real bacteria. Next, reproduce and scale the chemistry, then test the antibiotic against resistant bacteria. Lichirui Zhong, Richard A. Friesner, Edward B. A. Miller, and João P. Glm. Rodriguez benchmarked the cofolding model bolts on unseen ligand-bound human GPCR families and found that it generalizes reasonably well to receptor backbone placement but remains inconsistent on ligand pose prediction. The result is significant because GPCR drug discovery is a test case for whether AI can shift from pattern recognition to usable molecular design. The paper suggests an intelligence inversion in which models can now infer much of the structural scaffold, but the remaining failure modes show that disease treatment is still an engineering problem of reliable binding geometry, not just better prediction. If confirmed, this could help drug teams test better molecular ideas sooner instead of searching blindly. The useful part is a smarter starting point, not a finished medicine. Biology still gets the final vote. Next, show with independent benchmarks and laboratory work that the targets produce better drug candidates. The practical read is simple. These are signals, not promises. But each one is a concrete target that researchers, clinicians, engineers, funders, and the people most affected can connect to a next step. Keeping the evidence visible is how the Cure protocol stays current without pretending the work is finished.